Hello everybody, Kobe here for Science Sapiens, and today we're talking about DNA technology and gene regulation. A lot of science and science class talks about how all this stuff works, how DNA works, how cells work. But what they don't talk about is how people know they work, and this is what we're going to talk about today. This is how scientists, the processes science do to study cells and DNA today, and also we'll talk about how the cells themselves regulate their DNA. As always, this isn't meant to replace a class. It's just meant to be review, meant to be something interesting, great for studying for the SATs or a test or something, but don't let this replace your class. All right. So, DNA technology, just a brief overview of like why we care, what's the point. So what this picture shows here, it shows how we make insulin. So insulin is a hormone that the human body makes and needs, and diabetics don't make it properly. And they need to take injections of insulin to stay healthy. So what scientists can do is they can take the human DNA, they can isolate the gene for insulin. That's what they're doing here. And what they then do is they insert that into a piece of bacterial DNA called a plasmid. They break off the insulin gene out of the human genome, glue it into the bacteria genome, which is called recombinant DNA, so DNA that's been combined. And then they put that back in the bacteria, and what happens is the bacteria will start making insulin, and we can take the insulin from the bacteria, inject it, and keep diabetics happy. So that's kind of why DNA technology is important. Just one example of what it can do. And scientists use it all the time. All right. So there's a bunch of DNA technology. I'm just going to go quickly through each one, and then we'll get into a few examples and talk about what they do and how they work. So genetic engineering, brand new topic, isn't well understood. We're not very good at doing it yet, but what it is, it's the direct manipulation of genes. So basically taking a gene, switching the bases so it does stuff we want it to do. And again, very, very new, very unreliable, still kind of understanding how it works, what to do, but basically rewriting genetic code. Recombinant DNA is, like I said earlier, just DNA from two sources. So we take one species A, species B, stick their DNA together. That's called recombinant DNA. And it's actually easier to do than you think. Restrictions, enzymes are enzymes that are found naturally in cells, and they cut DNA in very specific sequences. So they recognize the sequence and they cut it there just like a pair of scissors. And cells in nature use this for protection. If a virus comes in, or foreign DNA comes in, and the cell doesn't recognize it, they'll just cut it to pieces. But what scientists can do is they can find enzymes that cut just where they want, and therefore cut out segments of DNA, which are really helpful for them. PCR, or polymerase chain reaction, is used every day, thousands of times a day, by scientists around the world. And what it does, it's an artificial process that copies DNA really quickly. So it's basically artificial replication. So an example of when this is used is at a crime scene. If they find a little bit of blood and they want to know whose blood it is, there's not enough DNA in there to read it. But what they can do is they can take the blood, take the DNA out of it, copy it 1 billion times, and then they can do whatever experiment they want with it. They can isolate whose it is. They can see, all that kind of stuff. So polymerase chain reaction is used to copy DNA very quickly, very efficiently, and in a lab. And then finally, a last process that's pretty old and is used all the time is gel electrophoresis. And basically what it does is it separates DNA based on size. And again, this is used at crime scenes too. So why do we care? It allows scientists to isolate, replicate DNA. It's used by pharmaceuticals all the time to make insulin, right? That initial example. There's also the ability that we can program bacteria to clean up oil spills, to take carbon out of the atmosphere, that kind of stuff. So we can change these little cells, these bacteria, to do really big processes. They might be able to make fuel for us one day. We don't have to, rely on fossil fuels. So it's all of these things that DNA technology could do one day. And then a buzzword you hear all the time is genetically modified organism. What is a GMO? And it's just any food source, any organism that has been modified by humans. So people today are all worried, oh, GMOs aren't healthy, we shouldn't be messing with the environment. Everything you eat is a GMO. Every single thing you eat. Corn naturally doesn't look like corn we eat. Thousands of years ago, millions of years ago, prehistoric humans took the best corn, the corn that was the biggest, the juiciest, the healthiest, and they ate that. And then they fertilized it, and they had a whole new crop of corn. And then they took the best corn, ate that, fertilized it. So GMOs, any food that humans have ever interacted with, really are GMOs. So it's kind of a silly concern that people have, and a lot of people who talk about it don't understand what it means. It just means that the organism has somehow been affected by humans. So keep that in mind, when you go to the supermarket, it says no GMOs. Well, it's not really that big of a deal most of the time. All right. So like I said earlier, restriction enzymes cut DNA at very specific points. And let's just take a look at this bottom diagram here. Here we have molecule A and molecule B. So two segments of DNA that are basically identical, right? And we want to stick them together. How do we stick them together? Let's say this is from human, right? This is the human insulin gene, and this is the bacteria. So I found a segment that's very similar, and I want to stick them together. How do I do that? So I take a restriction enzyme that cuts at a very specific place. It cuts when it sees this sequence, what I've just circled. Oops. Sorry, I messed that up. When it sees this sequence, it cuts it. Okay? Same over here. There's a second enzyme that cuts when it sees this sequence. So scientists will put the human DNA in a test tube, stick the restriction enzyme in there, mix it all up, and we'll get a ton of these cut pieces. And then they'll do the same with the bacteria. Put them in a different test tube, put a different enzyme in, cut them, and we'll see that we end up with these sticky ends. And what it means by sticky ends is that all of these exposed nucleotides, they want to bind with somebody, right? They're like, hey, I want a friend. They want to bind with somebody. And when these are introduced, when we put the two genes or the DNA pieces together, they stick, just like we see here. They anneal, we can cool them down, and then we get recombinant DNA. We see this new piece of DNA binds together right here, right? Same thing is shown over here, right? We have these two segments. We want to cut them. We get these sticky ends when we get their very specific restriction enzymes, and then they stick together. So it's a great way to connect two pieces of DNA. Okay, found naturally. So they have the site that they recognize is called the recognition sequence, or the recognition site, so that's where they cut. Sticky ends are the single strands. And then restriction fragments are just this fancy name for the pieces of DNA that the restriction enzymes make. So if I have a piece, cut them into two with a restriction enzyme, those two pieces are then called restriction fragments. And it's used all the time to make recombinant DNA. Cool. Now we're going to talk about gel electrophoresis. So what we have here is we have this blue thing here is a gel. It's kind of like Jell-O. But what it is it's porous. It has holes in it. And then what we do is we basically put electrodes. We put a negative charge here and a positive charge here. And then we put our samples of DNA into these wells. And we've cut these DNA samples with restriction enzymes, so they're very specific lengths. And we know the lengths of these restriction enzymes, or we can figure them out. And what we do is we put this DNA that's cut up into all these pieces into the gel. And we have these two electrodes, and we run the electricity, negative to positive like this, and the DNA is pulled through the gel. Remember, it has holes. So bigger pieces of DNA are going to have a harder time moving through, so they're going to move less far. So the larger fragments, the larger pieces are going to be here, and the smaller pieces will be able to be pulled through more easily, and they'll end up lower down. So big to low. And the reason they get pulled is because DNA is slightly negatively charged, very slightly. And what that does is it gets attracted to the positive end, which is at the bottom, and it gets pushed away from the negative end. So the electricity pushes it down. Example of when this is used is in a forensic lab. So let's say I commit a crime. I leave some skin at the crime. The police pick up the skin. They cut the DNA very specifically into enzyme with restriction enzymes into these restriction fragments and run it through a gel. And what they're going to do is they're going to cut it at places that people have a lot of diversity at. So there's these pieces of DNA, very short strands, that basically every person on the planet has a different strand, a different sequence. So when they cut my genes, or cut my DNA, it's going to make a very specific pattern, right? It's going to be cut because I only have some of these places where the restriction enzyme is going to cut, because these segments are so highly varied, I'm going to get a very specific pattern. And if they then bring me in for questioning, take a blood sample, take the DNA, and they find a pattern that matches, they know I was at the crime scene. Okay? So this is really important for isolating parts. So for example, if scientists took a human genome, cut for the insulin gene, they say, oh, I know the insulin gene is 100 base pairs long. I can run this sample through the gel with a marker that says, oh, right here, this length is 100. So now all of these right here are the insulin genes. So what they do is they just take an X-Acto knife, cut out the gel, melt the gel back down, and they have just the gene for insulin. So it's very important, used all the time. So let's just go what? Different DNA fragments cut, pulled through. Why allows us to isolate genes, and how, right? DNA is charged, so we can pull it with electricity. All right, PCR. PCR is kind of a hard concept to get your head around, but basically what it is, it's artificial replication. Really, basically, it's just artificial replication. Oops, sorry. So let's look here. Let me just get my laser pointer. Come on. All right. So if this is our initial piece of DNA, and this dark blue area right here is the area we want to copy, what we first do, excuse me, we denature the DNA. We break it apart, and we do this by heating it up. So we break the two pieces apart by heating it up to 90 degrees Celsius, really high. Then what we do is we add in primers. And if you don't know what primers are, check out my video on molecular genetics and the central dogma of molecular biology. So we add in primers, right? The primers stick to the start sequences, and we cool it down. And when this happens, we can start extending the sequences, basically elongating them, right? We have DNA polymerase called Taq polymerase that is meant to be used at high temperatures. So it'll bind to the primers. And remember, we're putting in primers for the genes we want to replicate, only those. And the polymerase will come in and copy. So then what we'll do is we'll heat it up again. Now we have two strands of the gene we want. We'll heat it up again, splitting it into two. Add more primers in, and then cool it down again and copy it again. And we'll just do this over and over and over and over again. It's cyclical. And basically what we do is we get an exponential growth. So if we start with one piece, then we get 2, then we get 4, then we get 8, then we get 16, right? We just keep growing and growing and growing and growing exponentially fast. So within a matter of hours or days, we can have hundreds of thousands of, or even millions or billions of pieces of this gene we want. And again, all we're doing is breaking the DNA apart, adding in primer, and extending the sequence of genes we want. So it's amplification. It's the replication of a gene or DNA fragment. Why? It allows us to take DNA and put it into a vector. A vector is an organism that carries DNA. Or it allows us to just get lots of copies so we can do lots of experiments with it. And it uses Taq polymerases, which is it's a DNA polymerase that's from an organism that lives in really hot environments. And because of this, the protein doesn't denature. It actually works better when it's hot. So the DNA gets hot enough to break apart denature, but the protein doesn't denature, and it can help, or it'll basically copy the DNA and replicate it. All right, so that's the DNA technology we're going to talk about today. Now we're going to talk about gene regulation really quickly. And what gene regulation is, it's how cells control what genes they want to regulate, what genes they want to express. So, for example, I don't need to, if I'm a bacteria, I don't always need to make a protein that digests food. If I'm not eating, it's a waste of my energy to make a protein that digests food. So I only want to turn that protein on when I have food. So that's basically what gene regulation is. It's saying when and where to make proteins. Okay, so transcription and translation all are regulated. So when they're on, when they're off, regulated by a cell. And a great example of this is cell differentiation. So every cell in my body has the same DNA, the same exact DNA. But my eyes don't need to express the genes for muscles, and my muscles don't need to express the genes for eyes. So my eye cells only express eye genes, and my muscle cells only express muscle genes. And in this way, I can get cell differentiation. I have cells that do very specific jobs. It takes a lot of energy to make a protein, so cells want to use as little energy as possible, so they only make proteins when they need them. And finally, we're going to talk about prokaryotic gene regulation, because eukaryotic gene regulation is really, really complex, and scientists are only starting to understand it now. All right, so they use something called Operons for gene regulation, and it's basically an on-off switch. So we're just going to go through some vocab before we look at the diagram. So an operon is the prokaryotic gene in the control elements. The promoter is the region of DNA where the polymerase binds. The operator is where the DNA can get basically turned on or off. It's the axis, the door to the DNA. And then we have something called repressors, which are proteins that stop DNA or RNA polymerase from binding, so we can't go through transcription. And then oppositely, we have inducers, molecules that bind to repressors and prevent them from stopping DNA. So they basically turn transcription on. And finally, we have co-repressors, so molecules that help activate or inactivate inducers or repressors. So these are probably not biological. They might be vitamins, minerals, stuff like that, that either, or they could be proteins, but they bind to the inducers and repressors to help them or turn them off. So let's look at this operon. It's called the Lac Operon. Lac standing for lactose. So basically, bacteria don't usually have the lactose sugar to eat, but when they do, they need to break it down. So most of the time, this gene is off, but it's inducible, meaning I can turn it off. So normally it's off, it doesn't turn on. To turn it on, I need or most of the time the repressor is binded on. The repressor is this red thing here. It binds on right here, and the DNA polymerase can't get by. It's like an off switch. But when lactose is present, lactose binds to the protein that turns the cell off, and it stops it from binding. It turns it into an inactive repressor. So we go along, we make the proteins that digest lactose, and we eat it all up. And when we eat it all, what happens? Well, we get rid of the lactose that's stopping the repressor. So the repressor binds back on, and we stop making the proteins that we need to make lactose. So when we don't have lactose, we don't make it. When we do have lactose, we make it. So this is inducible. It can be turned on, but most of the time it's off. This is opposite to the tryptophan, the TRP Operon, which is repressible. So most of the time it's on, but we can turn it on. Tryptophan is an amino acid that bacteria can make, or they can ingest. But most of the time they're gonna make it. So we have this repressor, right, which doesn't turn on normally. So normally we're going along, we're making tryptophan. We're making this molecule. But when we eat tryptophan, when the bacteria eats tryptophan, they don't need to make it anymore. They can just use the stuff they've eaten. So the tryptophan will bind to the repressor, turning it on. So it's a co-repressor. Binds, and then the RNA polymerase, this protein here, doesn't make the RNA to code for tryptophan. But when we, again, don't have tryptophan, when it's all used up, it falls off of the repressor. The repressor gets turned off, and we go ahead and make tryptophan normally. Okay? Again, remember, those are bacterial operons. So that is gene regulation and biotechnology. Hope you guys found it interesting and helpful. Leave your questions in the comments below. Don't forget to rate and subscribe. And as always, keep questioning.